வணக்கம். Welcome to NCERT Biology reading. In Biology, Class 11, Chapter 10, Cell Cycle and Cell Division. Are you aware that all organisms, even the largest, start their life from a single cell? You may wonder how a single cell then goes on to form such large organisms. Growth and reproduction are characteristics of cells. Indeed, of all living organisms, all cells reproduce by dividing into two, with each parental cell giving rise to two daughter cells each time they divide. These newly formed daughter cells can themselves grow and divide, giving rise to a new cell population that is formed by the growth and division of a single parental cell and its progeny. In other words, such cycles of growth and division allow a single cell to form structure consisting of millions of cells. 10.1 Cell cycle. Cell division is very important process in all living organisms. During the cell division of a cell, DNA replication and cell growth also take place. All these processes, that is cell division, DNA replication and cell growth, hence have to take place in a coordinated way to ensure correct division and formation of progeny cells containing intact genome. The sequence of events by which a cell duplicates its genome, synthesis, and the other constituents of the cell and eventually divides into two daughter cells is termed cell cycle. Although cell growth in terms of cytoplasmic increase is a continuous process, DNA synthesis occurs only during one specific stage in the cell cycle. The replicated chromosomes DNA are then distributed to daughter nuclei by a complex series of events. During cell division, these events are themselves under genetic control. 10.1.1 Phases of cell cycle. A typical eukaryotic cell cycle is illustrated by a human cells in culture. These cells divide once in approximately every 24 hours, figure 10.1. However, this duration of cell cycle can vary from organism to organism and also from cell type to cell type. Yeast, for example, can progress through the cell cycle in only about 90 minutes. The cell cycle is divided into two basic phases. One is interphase, another one is M phase, that is mitotic phase. The M phase represents the phase when the actual cell division or mitosis occurs, and the interphase represents the phase between two successive M phases. It is significant to note that in the 24-hour average duration of cell cycle of a human cell, cell division proper lasts for only about an hour. The interphase lasts more than 95% of duration of cell cycle. The M phase starts with the nuclear division corresponding to the separation of daughter chromosomes, that is karyokinesis, and usually ends with the division of cytoplasm cytokinesis. The interface, though called the resting phase, is the time during which the cell is preparing for division by undergoing both cell growth and DNA replication in orderly manner. The interface is divided into three further phases. One is G1 phase, gap 1, or S phase, L or synthesis, G2 phase or gap 2. G1 phase corresponds to the interval between mitosis and initiation of DNA replication. During G1 phase, the cell is metabolically active and continuously grows but does not replicate its DNA. Does not replicate its DNA. S or synthesis phase marks the period during which DNA synthesis or replication takes place. During this time of amount of DNA per cell doubles. If the initial amount of the DNA is denoted as 2C, then it increases to 4C. However, there is no increase in the chromosome number if the cell had diploid or to a number of chromosomes at G1. Even after S phase, the number of chromosomes remains the same. In animal cells, so please note here, how do plants and animals continue to grow all their lives? Do all cells in plants divide all the time? Do you think all cells continue to divide in all plants and animals? Can you tell the name and the location of the tissues having cells that dividing all their life in higher plants? Do animals have similar meristematic tissues? In animal cells, during the S phase, DNA replication begins in the nucleus and the centriole duplication is in the cytoplasm. During the G2 phase, proteins are synthesized in preparation for mitosis while cell growth continues. Some cells in the adult animals do not appear to exhibit division, example heart cells, and many other cells divide only occasionally as needed to replace cells that have been lost because of injury of cell death. These cells that do not divide further exist G1 phase to enter an inactive stage called quiescent stage G0 of the cell cycle. Cells in this stage remain metabolically active but no longer proliferate unless called on to do so, depending on the requirement of the organism. In animals, mitotic cell division is only seen in diploid somatic cells against this. The plants can show mitotic division in both haploid and diploid cells. From your recollection of examples of alternation of generation in plants, chapter 3, identify plant species and storage and stages at which mitosis is seen in haploid cells. 10.10.2 M phase. This is the most dramatic period of the cell cycle, involving a major recognition of virtually all components of the cell, since the number of chromosomes in the presence in the parent and progeny cells in the same. It is also as equational division, though for convenience mitosis has been divided into four stages of nuclear division. It is very essential to understand that cell division is a progressive process and very clear cut lines cannot be drawn between various stages. Mitosis is divided into following four stages: prophase, metaphase, anaphase, and telophase. 10.2.1 Prophase. Prophase, which is the first stage of mitosis, follows the S and G2 phase of the interphase. In the S and G2 phase, the new DNA molecules formed are not distinct but intertwined. Prophase is marked by the initiation of condensation of chromosomal material. The chromosomal material becomes untangled during the process of chromatin condensation. Figure 10.2a. The centriole, which had undergone duplication during a phase of interphase, now begins to move towards opposite poles of the cell. The completion of prophase can thus be marked by the following characteristic events. Chromosomal material condenses to form compact mitotic chromosome. Chromosomes are seen to be composed of two chromatids attached together at the centromere. Initiation of the assembly of mitotic spindle, the microtubules, proteinaceous components of the cell cytoplasm help in the process. Cells at the end of the prophase, when viewed under the microscope, do not show Golgi complex, endoplasmic reticulum, nucleolus, and the nuclear envelope. 10.2.2 Metaphase. The complete disintegration of the nuclear envelope marks the start of the second phase of mitosis, hence, the chromosomes are spread through the cytoplasm of the cell. By this stage, condensation of chromosome is completed and they can be observed clearly under this microscope. This then is the stage at which morphology of chromosome is most easily studied. At this stage, metaphase chromosome is made up of two sister chromatids, which are held together by the centromere. Figure 10.2b. Small disc-shaped structure at the surface of the centromeres are called kinetochores. This structure serves as the site of attachment of spindle fibers formed by the spindle fibers to the chromosomes that are moved into position at the center of the cell. Hence, the metaphase is characterized by all the chromosomes coming to lie at the equator with one chromatid of each chromosome connected by its kinetochore to spindle fibers from one pole and its sister chromatid connected by its kinetochore to spindle fibers from the opposite pole. Figure 10.2b. The plane of alignment of the chromosome at metaphase is referred to as the metaphase plate. The key feature of metaphase are spindle fibers attached to kinetochores of chromosomes. Chromosomes are moved to spindle equator at get aligned along metaphase plate through spindle fibers to both poles. 10.2.3 Anaphase. At the onset of anaphase, each chromosome arranged at the metaphase plate is split simultaneously and the two daughter chromatids, now referred to as chromosomes of the future daughter nuclei, begins their migration towards the opposite poles as each chromosomes move away from the equatorial plates. The centromere of each chromosome is towards the pole and hence the leading edge, with the arm of the chromosome trailing behind. Thus, anaphase stage is characterized by the following key events. Centromere split and chromatids separate. Chromatids move to opposite poles. 10.2.4 Telophase. At the beginning of the final stage of mitosis, that is, telophase, the chromosomes that have reached their respective poles decondense and lose their individuality. The individual chromosomes can no longer be seen in chromatin material, tends to collect in a mass in the two poles. Figure 10.2d. This is the stage which shows the following key events. That is, 1. Chromosomes cluster at opposite spindle poles and their identity is lost as discrete elements. Nuclear envelope assembles around the chromosomes cluster. Nucleolus, Golgi complex, and endoplasmic reticulum reform. 10.2.5 Cytokinesis. Mitosis accomplishes not only the segregation of duplicated chromosomes into daughter nuclei, karyokinesis, but the cell itself is divided into two daughter cells by a separate process called cytokinesis, at the end of which cell division is complete. Figure 10.2c. In an animal cell, this is achieved by the appearance of a furrow in the plasma membrane. The furrow gradually deepens and ultimately joins in the center, dividing the cell cytoplasm into two. Plant cells, however, are enclosed by a relatively inextensible cell wall, therefore, they undergo cytokinesis by a different mechanism. In plant cells, wall formation starts at the center of the cell and grows outward to meet the existing lateral walls. The formation of the new cell wall begins with the formation of a simple precursor called the cell plate. They represents the middle lamella between the walls of the two adjacent cells. At the time of cytoplasmic division, organelles like mitochondria and plastid get distributed between the two daughter cells. In some organisms, karyokinesis is not followed by cytokinesis, as a result of which multinucleate condition arises, leading to the formation of syncytium. Example: liquid endosperm in coconut. 10.3 Significance of mitosis. Mitosis or the equational division is usually restricted to the diploid cells only. However, in some lower plants and in some social insects, haploid cells also divide by mitosis. It is very essential to understand the significance of this division in life of an organism. Are you aware of some examples where they have studied about haploid or diploid insects? Mitosis usually results in the production of diploid daughter cells with identical genetic complement. The growth of multicellular organisms is due to mitosis. Cell growth results in disturbing the ratio between the nucleus and the cytoplasm. It therefore becomes essential for the cell to divide to restore the nucleocytoplasmic ratio. A very significant contribution of mitosis is cell repair. The cells of the upper layer of the epidermis, cells of the lining of the gut, and blood cells are being constantly replaced. Mitotic division in the meristematic tissue, the apical and the lateral cambium result in continuous growth of plants throughout their life. 10.4 Meiosis. The production of offspring by sexual reproduction include the fusion of two gametes, each with a complete haploid set of chromosomes. Gametes are formed from specialized diploid cells. This specialized kind of cell division that reduces the chromosome number by half result in the production of haploid daughter cells. This kind of division is called meiosis. Meiosis ensures the production of haploid phase in the life cycle of sexually reproducing organism, whereas fertilization restores the diploid phase. We come across meiosis during gametogenesis in plants and animals. This leads to the formation of haploid gametes. The key feature of meiosis are as follows. Meiosis involves two sequential cycle of nuclear and cell division called meiosis 1 and meiosis 2, but only a single cycle of DNA replication. Only a single cycle of DNA replication. Meiosis 1 is initiated after the parental chromosome have replicated to produce identical sister chromatids as the S phase. At the S phase. Meiosis involves pairing of homologous chromosomes and recombination between them. Four haploid cells are formed at the end of the meiosis 2. Meiotic events can be grouped under the following phases. Meiosis 1 is grouped as prophase 1, metaphase 1, anaphase 1, telophase 1. And meiosis 2 is divided into prophase 2, metaphase 2, anaphase 2, and telophase 2. 10.4.1 Meiosis 1. Prophase 1. Prophase of the first meiotic division is typically longer and more complex when compared to prophase of meiosis. It has been further subdivided into the following five phases based on the chromosomal behavior, that is leptotin, zygotin, pachytin, diplotin, and diakinesis. During leptotin stage, the chromosome became gradually visible under the light microscope. The compaction of chromosome continues throughout leptotin. This is followed by the second stage of prophase is called zygotin. During this stage, chromosomes start pairing together and this process of association is called synapsis. Such paired chromosomes are called homologous chromosomes. Electron micrographs of this stage indicate that chromosome synapsis is accompanied by the formation of the complex structure called synaptonymal complex. The complex formed by a pair of synapsed homologous chromosomes is called bivalent or tetrad. However, these are more clearly visible at the next stage. The first two stages of prophase 1 are relatively short-lived compared to next stage, that is pachytin. During this stage, bivalent chromosomes now clearly appears as tetrads. This stage is characterized by the appearance of recombination produces the site at which crossing over occurs between non-sister chromatids of the homologous chromosomes. Crossing over is the exchange of genetic material between two homologous chromosomes. Crossing over is also an enzyme mediated process, and the enzyme involved is called recombinase. Crossing over leads to recombination of genetic material on the two chromosomes. Recombination between homologous chromosomes is completed by the end of pachytin, leaving the chromosome linked at the site of crossing over. The beginning of diplotin is recognized by the dissolution of the synaptonymal complex and the tendency of the recombined homologous chromosomes of the bivalent to separate from each other except at the site of crossovers. These X-shaped structures are called chiasmata. In oocytes of some vertebrates, diplotin can last for months or years. The final stage of meiotic prophase 1 is diakinesis. This is marked by the terminalization of chiasmata. During this phase, the chromosomes are fully condensed and the meiotic spindle is assembled to prepare the homologous chromosomes for separation. By the end of diakinesis, the nucleolus disappears and the nuclear envelope also breaks down. Diakinesis represents the transition to metaphase. Metaphase 1, the bivalent chromosomes align in the equatorial plates. Figure 10.3, the microtubules from the opposite poles of the spindle attached to the pair of homologous chromosomes. Anaphase 1. The homologous chromosomes separate while sister chromatids remain associated at their centromeres. Figure 10.3. Telophase 1. The nuclear membrane and the nucleolus reappear. Cytokinesis follows and this is called dyad of cells. Figure 10.3. Although in many cases the chromosomes do undergo some dispersion, they do not reach the extremely extended state of the interphase nucleus. The stage between the two meiotic division is called the interkinesis and is generally short-lived. Interkinesis is followed by prophase 2, a much simpler prophase than prophase 1. 10.4.2 Meiosis 2. Prophase 2 Meiosis 2 is initiated immediately after cytokinesis, usually before the chromosomes have fully elongated. In contrast to meiosis 1, meiosis 2 resembles a normal mitosis. The nuclear membrane disappears by the end of prophase 2. Figure 10.4. The chromosomes again become com- and the chromosomes again become compact. Metaphase 2. At the stage, the chromosomes align at the equator and the microtubules form opposite poles of the spindle get attached to the kinetochores. Figure 10.4 of sister chromatids. Anaphase 2. Anaphase 2, it begins with the simultaneous splitting of the centromere of each chromosome, which was holding the sister chromatids together, allowing them to move toward opposite pole of the cell. Figure 10.4. Telophase 2. Meiosis ends with telophase 2, in which the two groups of chromosomes once again get enclosed by a nuclear envelope. Cytokinesis follows, resulting in the formation of tetrad of cells, four haploid daughter cells. Figure 10.4. 10.5. Significance of meiosis. Meiosis is the mechanism by which conservation of specific chromosomes number of each species is achieved by across generations to sexually reproducing organisms. Even though the process per se paradoxically results in reduction of chromosome number by half, it also increases the genetic variability in the population of organisms from one generation to the next. Variations are very important for the process of evolution. Summary. According to the cell theory, cells arise from pre existing cells, omnicellulae, ecellulae. The process by which this occurs is called cell division. Any sexually reproducing organism starts its life cycle from a single cell called zygote. Cell division does not stop with the formation of the mature organism but continue throughout its life cycle. The stage through which a cell passes from one division to the next is called the cell cycle. Cell cycle is divided into two phases called interphase, a period of preparation for cell division, and mitosis, M phase, the actual period of cell division. Interphase is further subdivided into G1S and G2 phase. G1 phase is the period when the cell grows and carries out normal metabolism. Most of the organelle duplication also occurs during this phase. S phase makes the phase of DNA replication and chromosome duplication. G2 phase is the period of cytoplasmic growth. Mitosis is also divided into four stages, namely prophase, metaphase, anaphase, and telophase. Chromosome condensation occurs during prophase. Simultaneously, the centrioles move to the opposite poles, the nuclear envelope and the nucleolus disappear, and the spindle fibers start appearing. Metaphase is marked by the alignment of chromosomes at the equatorial plate. During anaphase, the centromere divide and the chromatids start moving towards the two opposite poles. Once the chromatids reach the two poles, the chromosomal elongation starts. Nucleotides and the nuclear membrane reappear. This stage is called the telophase. Nuclear division is then followed by the cytoplasmic division and is called cytokinesis. Mitosis, thus, is the equational division in which the chromosome number of the parent is conserved in the daughter cell. In contrast to mitosis, meiosis occurs in the diploid cells, which are distinct to form gametes. It is called the reduction division since it reduces the chromosome number by half while making the gametes. In sexual reproduction, when the two gametes fuse to chromosome number is restored to the value in the parent. Meiosis is divided into two phases. Meiosis 1 and meiosis 2. In the first meiotic division, the homologous chromosomes pair to form bivalents and undergo crossing over. Meiosis 1 has a longer prophase, which is divided further into five phases. These are leptotin, zygotin, packaging, diplotin, and diakinesis. During metaphase 1, the bivalents arrange on the equatorial plate. This is followed by the anaphase 1, in which homologous chromosomes move to opposite pole with both chromatids. Each pole receives half of the chromosome number of the parent cell. In telophase 1, the nuclear membrane and the nucleolus reappear. Meiosis 2 is similar to mitosis. During anaphase 2, the sister chromatids separate, thus, the end of meiosis, four haploid cells are formed. Thank you.